In this video, we will demonstrate how to set up an Invitrogen QuantiGene Plex assay in the Luminex Exponent software. For the purpose of this video, we will show you how to set up a QuantiGene Plex 80-plex panel and will reference the Exponent software for the Luminex 200 instrument. Please refer to instrument user guides for applications involving the Luminex FlexMap 3D, Luminex MagPix, and Luminex XMap IntelliFlex instruments. Exponent software settings for the QuantiGene Plex assay can be found in the QuantiGene Plex user guide. To begin, refer to the LX200 user guide to log in to the Exponent software. Start the Luminex Exponent program from the Start menu or the desktop shortcut. At the System's login screen, log in using the login instructions provided in the Instrument user guide. Refer to the Maintenance tab to ensure that the instrument is properly calibrated and verified using the necessary startup, calibration, and verification procedures by following the system initialization process as outlined by the Luminex Instrument user guide. To program the QuantiGene Plex assay protocol, select the Protocol tab in the top navigation panel and select Create New Protocol, which will take you to the settings in the left-hand navigation. Fill in your preferred name. As an example, use Human QuantiGene Plex Custom 80-plex. Fill in manufacturer as Invitrogen. The version number is set by default to 1. If you wish to edit an existing protocol, the software will require you to change the version number in order to save the changes. For the volume field, enter 100. Enable the timeout to 45 seconds. Do not enable the XY heater. Change the doublet discriminator setting to 5000 to 25,000. Change the bead type to Magplex. Ensure that the PMT setting is on default. For analysis type, select None, which should be the default setting. For MagPix and FlexMap 3D systems, you will be required to select the plate name, which references the specified probe height for the QuantiGene Plex plate. You will be able to save the probe height for the QuantiGene Plex plate during the probe height adjustment process on these instrument types. The recommended settings are also available in the QuantiGene Plex user guide. Select the Next button on the bottom right. In the Analytes tab in the left side navigation, select the bead regions or numbers by clicking the ones from the available bead map that apply for the assay. Refer to the product information sheet that comes with the QuantiGene Plex panel or Plex set to select bead regions or bead numbers. Each region or number associates to a specific target which can be different between custom panels. Once a bead number is selected, highlight the name in the table on the right and type in the target or gene name associated with this bead, according to the product information sheet for this custom panel. Repeat this step until all beads have been selected and named. In the table on the right, the units field can be left blank, and for the bead count, make sure it states 100. Do not select the option for total count. Select the Next button on the bottom right. Here, in the Plate Layout tab on the left side navigation, you will see a blank plate map. However, you will not define the plate map at this point. Instead, this is done for each batch that is run, since the plate map usually changes with each batch or experiment. Therefore, just click Save at this time. The assay protocol is now saved. To design an assay batch using a pre-made and saved protocol, select Batches in the top navigation pane, then select Create a new batch from an existing protocol. On the next page, in the Protocol subtab on the left side navigation, enter in a batch name of choice. For example, Experiment 1 Human QuantiGene Plex Custom 80-plex. Select and highlight the appropriate protocol from the list under Select a Protocol. In this case, select Human QuantiGene Plex Custom 80-plex as defined previously when setting up the protocol. Select the Next button on the bottom right. In the Plate Layout tab on the left side navigation, select the appropriate replicate count for each background and unknown. In this case, we are showing them in triplicate. Use the grouping drop-down to select the correct setting. For vertical duplicates, use the 111-222 setting. For horizontal duplicates, use the 123-123 setting. Then select the desired wells for the background wells and select background. Complete the same procedure for the unknowns by selecting the desired wells for the unknowns. Select unknown. If desired, you can use the table to the right of the plate map to either import a sample list or you can enter descriptions for each unknown one at a time. This step can also be completed in the QuantiGene Plex data analysis app on the Thermo Fisher Connect platform. Once the plate map is complete, select Save. The batch is ready to be read at this time, or it can be saved as a pending batch. To run the batch, open up the Batches tab in the top navigation pane and then select the pending batch that was just saved. Select Run to begin the acquisition process, which defaults to the current batch section in the Results tab. During the run, you can monitor different parameters in real time by reviewing the histograms, scatter plots, bead counts, and median fluorescent intensity values. For details on these parameters, refer to the Instrument User Guide. At the completion of the assay run in the Results tab, select Saved Batches in the left side navigation menu and pick the relevant batch. Here you have the option to open the batch via the Open button and review the raw data. Step-by-step analysis for QuantiGene Plex data will typically be completed in the QuantiGene Plex data analysis app on the Thermo Fisher Connect platform or manually in Excel. To export the data file, select Export Results.csv. Store the CSV file to a chosen folder so you can use it on the QuantiGene Plex data analysis app. Refer to the video titled How to Use the QuantiGene Plex Data Analysis app on the Thermo Fisher Connect platform for how to import the CSV files and use the analysis features. Refer to the Luminex Instrument User Guide to run the necessary shutdown procedure. Thank you for watching this video. For more information about the QuantiGene Plex assay, visit thermofisher.com/QuantiGene. And for more information about the QuantiGene Analysis app on Thermo Fisher Connect, visit thermofisher.com/QG Analysis app.